record. So just go ahead whenever you're ready.
Okay, so yeah, today I thought I'd just obviously run you through the outreach blog that I've been doing, which is kind of titled From African Clawed Frogs to Humans and How Do Cells Copy DNA. There we go. So what I thought I'd briefly go over is first of all just like introduce my own research and kind of what the focus of the blog is going to be about. Run through my sort of thought process of why I chose a blog and the planning behind it before kind of showing off a little bit what I've done and then future ideas of where I can go next, really. So about me, I'm a third year PhD student working in the chromosomal replication group in the dental medical school. My project itself is focused on understanding DNA replication. So DNA replication is a process that every cell uses to copy its genome once every cell cycle. It's really important that it's really tightly regulated and carefully controlled, because if there are any mistakes, then the cells are going to get damaged, and then that damage might lead to things like cancer developing in the future. So to do my work, I use a number of different systems, including bacteria, frogs, which is a little bit of a more unusual one, and human cells. So I kind of wanted to encapsulate all of that in what I wanted to achieve, but I think my overarching aim for the blog was to provide an activity or event that's suitable for, I was hoping for late secondary school age children, so 14 to 16, that are interested in either DNA replication, or generally wanting to learn more about life as a research scientist. Because I was thinking of myself at that age, I probably didn't know that much about what went on. So I thought if I could add little bits of that, it might help. So I then went on to decide, obviously, how to deliver the activity, and because we couldn't do it in person, that's have to switch to online. So I kind of came up with three options, which was a video activity or Zoom, a podcast, or an online blog or a website. So I kind of went through each one. So a video activity, I thought this was probably the most interactive way of doing it, because you can get direct discussion between people. But I did also think it might potentially limit the reach of the audience, because obviously you need everybody to be available at a set time for this to happen. And I've also actually done some video-based outreach stuff in the past, so I kind of wanted to try something a little bit different. Thought about podcasts, so I kind of thought podcasts were quite a nice light way of information delivery. You didn't need, say, a session attendance, so you had a wider audience reach. However, if there was no, I thought there was obvious podcasts, you're just listening to something, there's no direct way to show any visual aids to help put away, to give what you're, to explain what your figures, to explain what you're typing or saying, to like kind of help the reader understand. So I thought that was quite a big limitation, and I wanted to include certain visual aids for what I was doing. So I kind of settled on a blog. So yeah, I thought a blog was available anytime. You can importantly change it based on feedback, so I could develop it based on what readers thought. Easy to link resources, and you can, although it's harder, there is still the opportunity to provide feedback, to get questions for some sort, to start discussions. So yeah, I decided on a blog, and for a couple of reasons. Most of them are shown here. So first, you can think you got a wider audience reach. There's a possibility, obviously, which I quite like to do, I think, to follow it on or continue it, and you can develop and evolve the posts almost around what feedback you get. I also thought that it might be useful for myself as well. So our lab does quite a lot of, or some outreach things, so it might be useful as a resource we could provide there. And I'm also into Science Mentor, which I thought it might be useful again as a blog to maybe provide extra information about what I do during these sessions. So then we get onto the blog itself. So it's a WordPress hosted blog, and I've had it shared through the MDS outreach social media channels, and I'm arranging for it to go to the school's newsletter. So if I just click through and hope this works. Okay, I think that worked. So yeah, so this is the blog itself, got the homepage, a bit of information about what the blog is, and then I've got a link to a feedback form just for a bit of information if people wanted to fill that out. Then the blog itself, so it's got a picture of the lab, and then currently 3 posts that are up at the moment. One of sort of the visual techniques that we use in the lab, which I did an example of microscopy, an experiment to try it yourself, and an introduction to my sort of research field. So I thought I'd just go over 2 of the posts. I thought I'd leave the background information to my research field kind of out of the presentation, because it's more of just an information post and not that easy to present as such. So I thought I'd go for more interactive components. So the first of which was a DNA extraction. So I wanted to add some sort of experiment to do from home as a way to kind of make it a bit more interactive, have a bit more participation as such. And I know DNA extraction is probably the most common outreach experiment around at the moment, but it's also quite relevant for the work we do, we do this process very regularly, and it does kind of underpin a lot of the further work we do. And also, because I work on DNA, I thought it might be a good one to include, because whoever's doing it gets to see DNA at the end. So on the right, I've just included a PDF that I made for the blog just to show briefly how to do it. So on the left, I have just for fairly easy to follow instructions on how to do it. And then on the right, I just have a few comments of how the process that you do at home links to the process we do in the lab. So it can kind of put it into context kind of thing. And then, no, don't let me click. The other post I wanted to kind of go through is, so I thought about explaining kind of the science we do and the techniques we use, but I wanted to stay away from all of the core biochemical things because they're not that interesting to explain or to look at, to be honest. So I didn't think they'd be that well suited to a blog. So I kind of stayed to the more visual things, which led me to start with microscopy, because it was sort of the immediately nicest images to get hold of while I'm not allowed in the lab. So in this post, I've just briefly explained how it works, kind of the idea, methods behind it all, and then included these images, which are just normal cells, so somebody reading can actually have a look what a cell looks like under the microscope, and then different sites of damage, so potential sort of repercussions, of course, if DNA replication can't complete it normally or if something goes wrong. And then I just thought it was also quite a nice visual way of seeing the importance of the process on a whole. If it goes wrong, you can see these marks of damage appear, and then this leads to X, Y, and Z potentially down the line. So that's kind of the posts I have up on the blog at the moment. I was thinking now, ideally, if depending on time, obviously I'd like to update it potentially once every 2 to 3 weeks or quicker, but depends how shift work and currently things go. But a couple of things I thought I could include in the future. So one is to build on this idea of showing off visual techniques we use in the lab. So I thought 2 potential options is flow cytometry, which is the bottom left image here. So the graph is fairly complicated at this point, but it's just an example. I thought I'd need to properly think about how to present it properly. But I thought it's quite a nice way, because we can just see as DNA increases, so as DNA replication takes place, the level of the protein decreases, essentially. And then the figure on the right, which is definitely the more interesting technique in terms of, I think, especially in terms of outreach, because it's a lot more visual, is a new technique we're doing, which is single molecule imaging. So we can effectively use this to look at DNA and look at things happening live. So this image, all of these purple strands are actually single units of DNA, which we've stretched out under the microscope, and we can actually look at proteins binding and actual DNA expanding as it's replicated and things like that, which I thought might be cool to include a few sort of videos of that happening, because they're also quite nice to look at, really. But obviously that would be quite a more science heavy kind of side. So I wanted to carry on with the experiments to try at home as well. So obviously DNA extraction from strawberries is one, but I thought about some others we could potentially do, and I've come up with these three. So one is obviously you can make homemade Petri dishes and you can grow your own sort of bacteria from your hands, which is harmless generally, and it's really easy to do. And it also relates to work again that we do in the lab, because we do culture quite a lot of bacteria. Nowadays, you can get a lot of at-home sort of USB microscopes, which are fairly powerful for £5–10 on Amazon. So I thought it could be a good idea maybe to show how these work, buy one of these, show a good one that people could use if they were interested, and maybe look at how to prepare samples and look at this stuff from home. Or there's the sort of more difficult option, which is you can grow bioluminescent algae at home, which I thought might be good because you can also combine it with the microscope post if I did that to see that in a bit more detail. Or I'd also like to also add a couple of posts about what the research is like on a general day-to-day basis, because I thought that was probably the most unknown at that age, probably, like what is actually going on every day in the labs.
1 minute, Shaun.
Okay, I think I'm pretty much done. Yeah, so the only final thing I wanted to chat about was, obviously it's a blog, so I found starting a two-way conversation is important, but it's probably difficult on a website-based platform. So I've tried to include as many ways as getting in contact and asking questions as possible, which have included a contacts page with my science Twitter email or the built-in WordPress form, as well as a feedback form here, which was just on Google Forms to sort of help me judge the difficulty level I've set the content at, and whether what people like, what people don't like, to kind of adjust what's coming next. And then, yeah, I think that was pretty much it. So, yeah.
Perfect timing. 11 minutes 58.
Oh, brilliant.
Great. John, do you want to jump in first, or shall I?
I like it. I was a microbiologist for 10 years more. So for me, this is great. And you're right, outreach, it does happen a lot with DNA removing from strawberries and passion fruit and whatever people can lay their hands on, small children even. That's wrong. What is it you wanted to achieve throughout the whole thing of this? For you, what would success have looked like at the end of it?
So I think to kind of show the show that science, I think, anybody can do science, show that it's accessible and you can do it at home, or you don't have to be, say, a research scientist. So you could do all these little experiments at home and you can follow interests if you want. But then also at the same time, maybe encourage people to look more into things like DNA replication, bits like that, because it is, I'd say, a fairly small field. Like for what I do anyway, there's only 2 or 3 labs in the world that work on it. So it's a very niche area that I feel like not many people will have necessarily heard of.
It's easy, but everybody's got DNA.
Yeah, exactly. That's the thing. It's everybody's got DNA, but then
yeah, we've all got DNA. We don't all necessarily know how it works.
Yeah,
but we've all got it.
Yeah, no, yeah.
So yeah, I wondered about that as well with the content. I've got a couple of questions that I'm going to tag together. So just on that, did you think about how you might tell the story a little bit more of why you're doing these things? So it might not be immediately obvious to somebody that it's really, really hard to look at one individual cell and that it's taken a kind of leap of technology. So did you think about that at all? And did you think about how you could gauge any of that? Are there any ways you can think of that you might be able to embed really short responses into the content?
Yes, so in terms of, I'll just stop the screen so I can see a bit better. In terms of like how the technologies progress, I did think about doing that, and particularly I thought if I was going to follow up on the, let's say, the line of going into what techniques we do in the lab, if I did a post on, let's say, the single cell stuff that we're now slowly getting into, I could go from the beginning and then kind of show where it's come along in a timeline and then go to the single molecule itself. And then, yeah, in terms of embedding the stuff, I think it would be a really good idea to do. Just provide a bit more information, you know, kind of show what's happening. I just need to think about exactly how would be the best way to do that.
Yeah, I just wondered whether you could use little polls or anything like that, where it's just a very quick, it's not as in-depth as your feedback form, but a bit of a sort of dipstick test of, did you know this, or did you know, do you, there might be things like that.
It would be, yeah, it's further down. I think because at the moment it's on the free version of the WordPress site, you can only really include links and things. So I think that is an option, but it's behind a paywall at the moment.
And then just, sorry, John, I'll let you back in, but
no, carry on.
Do you know anything about, at the moment, what your reach might be? And you've got some demographic questions, I didn't know. So have you had any feedback through your feedback form yet? Do you know how many hits you've got, that sort of thing? Do you know who's engaged? And going forward, what might you do? So will you, you've said you've made contact through the outreach teams, which is really, really good. So might there be some actions that you can follow up and try and get some concentrated feedback on the current content and maybe where you're going to go?
Yeah, so in terms of things at the moment, I think it's a little bit too early to say, because like I don't think the school's newsletter, for example, has gone out yet, which I think would be the big push. For social media things, I have seen increases in visits to the site. I mean, there's not massive, but I think I'm getting between 20 and 30 people a day. So it's okay, it's not hundreds, but I think that will hopefully increase when the schools go out, and then dependent on how the feedback things going in, it might be better to host some sessions, like one or individual sessions based on the content, and then see what people think, and then I can use that as a more direct feedback to kind of feed into the blog as about where that could go in the future. If that makes sense.
Yeah, I think it'd be fascinating to have some sort of focus groups, or where you can, maybe with classes, and you might try a couple of different age groups even, just to test your original thought of, oh, this will be the key age group. First of all, is that the key age group that responds? But what are their, if that is the key age group, is the content right?
Yeah, that makes sense.
John?
Yeah, actually, you'd already picked up two of the questions that I was going to ask.
Sorry.
One of them was about spread. Now is about sort of the stats that you were getting out of the WordPress engine, because I know on the free version you still get the ability to see how many people are hitting what pages. The other one was about what you were going to do taking this forward as well. And I know you talked quite a lot about the kind of things, the more microbiology, you know, grow your own slime mold, that kind of thing. That's kind of cool. For the moment, we also talked about going forward with this. I mean, we're sort of COVID-19. We have a lot of opportunities, but also a lot of challenges going forward about how we do that one-to-one engagement with people. And I think a lot of the school sessions that we would normally have run via outreach and things like that, they're not going to happen. So what would you do with that in mind to then step forward and go, okay, we are going to be doing virtual interactions. What would you bring to not just the website, but what else could you bring along with it, and how else would you tie the website into maybe a wider strategy to be able to deliver this in a way that could, you know, put you in a position where you're getting much more engagement?
Yeah, so obviously it's hard because we can't do anything in person, so it'd be easy to go somewhere and deliver something. But I did think, because I think maybe with the experiments kind of like that you could do on your own, it might be good to do them as like a live follow-on session, if that kind of makes sense, to help things go along. And then you can recruit certain age groups in, and then design, sort of tailor what you're doing in the session to the age group. But yeah, it's quite difficult apart from that, obviously, because it's all online. And then I feel doing it online is quite hard, because it's either like an information dump through a thing, and it's quite hard to process, and then I feel ask questions straight away. So I'm not 100% sure. I would like to do some more in-person things whenever we're allowed to, but obviously that's dependent on what's going on.
Sometimes I know what you mean. I'm definitely the sort of person that needs to be in front of an audience to really make something. I've assumed that we're not doing anything until next year, but that doesn't mean that I'm not going to do outreach. It means I've just got to be creative and do it in a different way. I was just thinking of Because as a former microbiologist, one of the things that I really loved was, it's not a microbiology thing, but do you remember the pitch drop webcam? There was basically this thick pitch tar, and it would slowly drop.
Okay.
They had a webcam on it, and it would move like, you know, a millimeter every year or something like that. And actually microbiology is great because you can set up and have a live growing webcam. Okay, here's our slime mold, it's been growing for 2 days now, what's happening, you know, those kind of things. They're the sort of things that bring people back in to see how it's going. And you sort of, it's like that old experiment where you grow peas in a jar on your windowsill and with, you know, blotting paper and stuff like that. I think those kind of things can sometimes be nice to hook people in, to bring them along with it. But they always need to know why they're doing this, why they're, you know, I think that's also good, because the other question that I had was about story. And if you were going to frame this as a story, are there elements that you could, I mean, when we talk about DNA, history's full of stories, but are there elements of story in there that you could bring out in the next one? It's kind of a rhetorical question, really. Yeah, I seem to have answered my own question. I'll shut up.
Yeah, I mean, it's quite a difficult one to answer, because I think I want to, because obviously there's so many resources out there on DNA itself. I want to kind of stay away from that, but then you can't stay away from it because it's a process involving DNA for the whole replication. But then, yeah, it's quite hard because at the moment, in terms of what's known, we don't know enough to make a story about everything scientifically. So then making that into like an outreach appropriate thing is hard. But I think I like the idea of like the time lapses of things dropping and things, because I could definitely do that. It just depends on obviously equipment, because it's all booked and then not booked and then you're not allowed to use it. But
yeah, there's always a corner of the lab you can grow slime mold in. You talked about DNA problems in replication and sort of things. What sort of disease states do you have in mind, particularly with your research, when it comes to the kind of things that you're focusing on?
Okay, so there's always cancer, which is a big one, but then that's just because any problem with DNA makes the genome unstable, which could potentially. But then in terms of more specific things, so the actual protein I'm working on, I haven't really mentioned it today, causes dwarfism when it's mutated. So there's that I could follow up on. So when that's mutated, you get overall DNA replication goes slower. And then that means the cells grow slower. So there's a lot less growth when you're developing. Mm-hmm. So it's kind of that I could follow down as well if I went more sort of disease linked kind of thing, to give it a bit of context about why the research is immediately relevant. Because I feel in a lot of research, if it's just say a biological process, it's hard to then put that into context. Well, why is that knowledge particularly necessary? If that makes sense.
I think those kind of things, I mean, you talk about DNA being a wide field, but in this case, it sounds as though there is an element of USP, because, I mean, just off the top of my head, like, the internet's a big place, but I'm not familiar with any website that tackles those two things together, DNA and dwarfism. So I think tactfully done, I think that could be a really interesting direction to take this forward. And obviously there is a very important group of stakeholders that would like to know more about that as well. Yeah.
I think actually you've got so many elements of story. It would be lovely to play around. I think to take a step back and see what people are fascinated by. I think people will be absolutely fascinated by, you skipped over the whole thing about toads. No, frogs, sorry, African clawed frogs. I'm sorry. But that's the sort of thing that in a scientific context and research context, we just go over the model organism. But a 16, 17-year-old will be like, why the poor frogs? And they will be really angry about the use of frogs. And so there's that but I think there's a few things that you could throw out there, that you've got also that metaphor about, we talk about DNA all the time, and that's really the set of, roads or whatever metaphor you want to use. What we don't think about is how are those roads built, maintained, and that's your proteins and everything else. And sort of, so I think actually what would be really nice is whether you could have, I get your point about doing a sort of Zoom session with a school or whatever, that you don't want to just tell them a lot of stuff and then say, right, what do you know about DNA? Tell me now. But I think there's a quite light conversation that you can have at quite a high level, just about what words they think of. You could do some word associations when you talk about DNA, and then to just give, well, did you know we use different DNA from different things? Because it allows us, you know, just some really light things, and then just get them to kind of vote or have a bit of a creative response, draw something, write something on what really they want to know more about. So I don't think you have to delve down, because actually you've got so much to choose from at that high level. You could take it so many ways.
I thought about doing things about the frogs, but I was a bit unsure, because obviously animal research is a little bit controversial, so I wasn't sure how to approach it. I mean, the plan is to do something along them lines, but it's just, I think it'd take me a good think to figure out how to write it tactfully, kind of thing.
Cancer Research UK have done some really good stuff, because they've really tried to take, particularly obviously they're looking at mouse models more than anything else, but they do have some stuff on other areas. But I think that thing of how do we know the things we know is really interesting to people. To say we just found this species of frog that's got these particular cells that make it possible we can do these experiments. I can't remember what it is that's specific about the frogs, but I remember when I did my PhD, I worked alongside Arga Gambus, who I think
she's super
led a lot of this stuff. So I remember about the frogs. But that sort of thing just isn't obvious to people at all. And for you to say, well, I'm not working on the DNA. Of course I'm not. I'm working on the protein. Just that concept. It's a whole ecosystem of stuff. And that's why we haven't figured it out yet. Is really nice to you can just take little tiny elements.
I think the potential to unpack these stories and controversial topics, health topics, is massive. And I know that you can't build an instant following like that. You are not Beyonce. You know, it's Shaun, I'm sure you're not Beyonce, but but I've lost my train of thought now. But actually, to keep these things forward, it is a case of just continuing them and letting them build up. I mean, you mentioned that you're part of your team are also doing the Into Science thing.
Yes.
Yeah, have you thought about widening this up as well, so it's not just specifically yours, but there are other people in the lab that will also be working on the same kind of model, the same specific methodology or something like that. Have you thought about opening them up and letting them contribute to it, and you curate you direct what they sort of do? You can chief editor and let them contribute to your website.
Yeah, it was an idea, but I haven't really approached it or gone any further than thinking it's a possibility to do, like other people can write a blog, or maybe some other people from different labs who work on similar things but slightly different things can but I hadn't got any further than thinking it could be a good idea.
It could. Well, it is a good idea.
Yeah.
It is, without sounding big-headed, it is a good idea.
Yeah.
And if ever you're stuck for something to say, I type, I don't know, if you type frogs or DNA into Google and then just hit news, most recent, you'll find out what's happening in the news stories about a particular topic, and that it means that once a week you're never short of a topic. You can contribute something, even if it's recipes for frog's legs. You know, appeal to a different audience on a different reason. There's always a way to hook people into the story. Oh, while we're here, let me tell you about why we're talking about frogs. Well, it's all to do with dwarfism and for instance, those kind of things. So, take time to build these things. These things do take time to build. But still, you're getting 30 to 40 hits a day, so that's very good.
Yeah. I think we should let you off the hook now because we've run over.
Yeah. Sorry, my bad.
That's all right.
Thanks ever so much for presenting your project and for the hard work that you put into it.
Yeah, it's been fun actually.
It's been great to see. Oh, I'm glad. And we look forward to see how it goes forward in future as well. I hope this module won't be the end of it. And as I said, I'll be in touch with any additional comments and all the rest of it to do with the assessment as well. All right.
Yeah, that sounds good.
Lovely.
Shaun.
Good to see you.
Perfect.
Thank
Thank
you.
you.
Bye.
Thank you.
Bye.
Bye.